Hello everyone, this is Shara. I'm Narval from BS Ed Sciences 3B. So I'm going to report about the Spirometra Mansoni from Kingdom Animalia, phylum Platyhelminthes, class Cystoda, and species S. Mansonoides. So my report focuses on characteristic, life cycles, disease and diagnosis, symptoms and treatments, and prevention and control. Spirometra mansoni. So what is Spirometra mansoni? Spirometra mansoni is a genus of pseudophyllid cestodes tapeworms with multiple segments and two or sucking grooves as adults that reproduce in canines and felines or domestic cats, but can also cause pathology in humans if infected. As an adult, this tapeworm live in the small intestine of its definitive host and produce eggs that pass with the animal feces. So let's go on to the characteristic. So these are the characteristic of Spirometra mansoni. Adult worm measures 60 to 100 cm in length. Scolex is elongated and spoon-shaped. It is characterized by the presence of two sucking grooves. It also consists about 1,000 proglottids. So proglottids means one of the segments of a tapeworm containing both male and female reproductive organs. The proglottids increase in size progressively as they become remote from the head of the worm. It also had two hosts. The primary hosts vary and include dogs and birds. The first intermediate host is copod or crustaceans, and the natural second intermediate hosts are usually birds, reptiles, or amphibians. So we humans usually contract the disease by consuming the raw flesh of one of the natural intermediate hosts, or more likely drinking water with copods. So an animal is small enough to go unnoticed in a glass of water. So an egg of Spirometra mansoni is approximately 52 to 78 micrometer times 31 to 44 micrometer. As you can see in the picture, the egg is light yellow in color, and it also a tape-like and segmented body in white color. So let's go on to the life cycle of Spirometra mansoni. Life cycle is quite complex with two intermediate hosts. Intermediate hosts are organisms that support or house the immature or non-reproductive form of parasite. Basically means where the parasite grows up. Definitive hosts are organisms that support or house the adult or sexually reproductive form of the parasite. This tapeworm name actually comes from the spiral-shaped uterus. Spiro means spiral and metra means uterus. So let's go on to the life cycle. So adult members of a genus Spirometra live in the intestines of dogs and cats. Eggs are shed in feces and embryonate in the environment. Then, eggs hatch in water and release choricidia, which are ingested by copods. The choricidia develop in a porcercoid larvae in the copod intermediate host. Then, the second intermediate host, including fish, reptiles, and amphibians, ingested or infected copods and acquire procercoid larvae. Procercoid larvae develop into a plerocercoid larvae in the second intermediate host. The cycle is complete when predator or a dog or cat eats an infected second intermediate host. Humans cannot serve as definitive hosts of Spirometra, but serve as a paratenic or second intermediate host and develop sparganosis. Humans acquire sparganosis by either drinking water contaminated with infected copods or consuming the flesh of an undercooked second intermediate or paratenic host. Spargana can live up to 20 years in the human host. So these are the disease or causes that we can get in Spirometra mansoni. First is drinking contaminated water that contains copods, or water that been contaminated with infected spirometer. Ingesting undercooked meat or raw foods such as seafoods or frogs, you can avoid getting it by way in cooking it thoroughly before eating it. And using contaminated meat as appetites are the main routes of infection. So these are the diagnoses for adults. Spirometra Mansoni infection. It is based on the demonstration of eggs in feces by microscopy. Then, after surgical removal of worms, diagnoses have been found. It's typically diagnosed following surgical removal of the worms. And biopsy with worms is another way to get a biopsy sample that has the worms already in it. In addition, to diagnose a Spirometra infection in humans, a serodiagnosis ELISA can be useful to target anti-Sparganum IgG antibodies within the blood. This diagnostic method is useful around 10 to 12 days post-infection and is almost 100% effective at detecting the anti-Sparganum antibodies in the 14 to 22 days post-infection. And serodiagnosis of sparganosis is a useful for early detection method. So these are the symptoms: vomiting, numbness, then itchiness and inflammation. This become itchy and inflamed, can develop anywhere on the body. The larvae migrate to different locations all over the body. Some of these location includes the tissue of the chest and eyes, and can also have a abnormal skin sensation. So these are the treatments. So in animals, it can be treated with Spirontel and Praziquantel. And this is according to my research. And also, the best way to treat the condition in human is with surgery. And accordingly, most drugs treatments in humans are unsuccessful at getting rid of larval stages. So let's go on to prevention and control. First is avoiding ingestion of contaminated water and making sure that meat that might contain sparganum is sufficiently cooked. Also, avoiding using the tissue frog of other cold-blooded animals for medical purpose. Infection of human may be prevented by avoiding eating undercooked frog or fish and avoiding drinking infected water. That would be all. Thank you.